morning, everybody. This is the Global Eye Genetic Consortium meeting. I'll be organizing this meeting with Dr. Jian Prakash and also with Dr. Natarajan and Dr. Kumar. And the first speaker is Dr. Kumar. Yes, please. And he will be talking about the contribution of India to global eye genetic consortium, genetics knowledge.
So good morning, everyone. At the outset, I would like to thank the organizers of AIOC to give me this opportunity, and also the key members of the GEGC, Dr. John Prakash, Dr. Natarajan, current president of AIOC, and Dr. Takeshi Iwata for this opportunity. So the next few minutes, I'm going to talk about the contribution made by several eye geneticists who have been primarily working on the laboratory side in India, and what was their contribution. And therefore, I'll start with the method, and this is the structure by which I'm going to give the talk today, and then I'll go through the regional contribution and so on and so forth as it's shown in the slide. Primarily, I retrieved data from the PubMed with certain keywords. And let's first see the SARC contribution. This is a slide that I used in the AEGC meeting in Colombo, which was organized by Dr. Jan Prakash. So what you see here is different countries from Afghanistan to Sri Lanka, which are SARC countries, and then comparing that with the research work in eye genetics done in the US. And the keywords used were country name, genes, disease, and 2 years in 2014 and 16. And you can see that in Bangladesh, when I put eye genes, then there are only 3 different papers so far that was obtained. This is 2016 data. And you can see in India that it's about 248 in 2014 for eye genes and 345 for 2016. And what you see in the bottom is that if I used the keyword eye genetics, okay. And if you compare that across several disorders, this is the data we have in India. And then if you compare that with the US, okay, so that's about more than 6,000 odd papers in 2014 and about 7,000 odd papers in 2016. And totally for eye genetics, you have nearly 17,948 publications that you can retrieve in PubMed. So nowhere you can compare ourselves in India, so which is one of the factors that we have to do more work to catch up with countries. Even with China, if you compare, we are not anywhere near today. Let's move on to the India's contribution. So again, I used the same method, and I used a few of the terms. So when I start from the lids onwards and then move towards the optic nerve and also diabetic retinopathy, these are the different publications that has been can retrieve in PubMed. Okay, and these are the institutions which contributed, and these are the significance. So starting from cornea, if you see that a good amount of work has been done in CHED, and primarily by Dr. Sundaresan's group in Arvind Eye Hospital. And if you look at cataract, and you look at primary congenital cataract, genetic point of view, then it is actually L.V. Prasad, which has done a lot of work. And so it is for glaucoma, congenital glaucoma, and people have worked quite a lot in this. Primary congenital glaucoma is the disease, in fact. And in LVP, quite a lot of work has been done compared to the other institutions. And these are all retrievable data. There might be some clinical information, but this is purely a genetic publications that I could retrieve. Now, retina is the maximum, about 200 odd papers that has been published by multiple institutions from Arvind, LVP. This is the Mohan MV Diabetes Center in Chennai, and of course, Narayan Nethralaya and the Shankar Nethralaya. Significant work was the discovery of RPE65 gene. Okay. And as far as AMD is concerned, Indrajit Kaur has done a lot of work in LV Prasad with the CFH marker that they found in similar to the validation that was there in other countries. Few publications have been done in diabetic retinopathy. At one point of time, there was more genetics and diabetic retinopathy paper in India compared to other countries. Then subsequently, the GWAS studies took over, and you have more publications from rest of the world, mostly in Europe and North America, that has been published. Now, let's see who are the people who have actually done these kind of contributions in India. So, who are they? So, these are the people. So, if you see iGenetics work, there are many places it might be done, but these are ones which we could retrieve out of publications in the PubMed. So, in Ahmedabad, you have the Ila Devi, where Dr. Vidya Nair and Dr. Rajkumar have been working. Now, Dr. Rajkumar has moved to Mumbai, Aditya Jyoth. And in Bangalore, at Narayan Nethralaya, you have Nalatambi, and he was a product of Arvind, and then he's now working at Narayan Nethralaya in Bangalore. Of course, Ram Prasad was my PhD student, and he's currently with a company called MedGenome, and he did quite a lot of work in iGenetics. Arka Subra Ghosh is a good scientist. He's working on gene therapy in Bangalore at Narayan Nethralaya. Vanirajan and Dr. Sundaresan are in Arvind, Madurai. And in Chennai, quite a lot of work was done in Shankar Nethralaya by Sri Priya, J. Madhavan, Saumitra. I couldn't get the photograph of Saumitra. Then Chitra, sorry. So Chitra, Indrajit Kaur and Shubho are from LV Prasad Eye Hospital. And Kunal Ray and his team were in Kolkata in ICIB. And of course, in Delhi, Radhika Tandon has been trying to work in ophthalmologist, and then, of course, Vanita Kumar in Amritsar. So these are the people who have really contributed to, I would say, about nearly 400 to 500 publications in iGenetics in India. The strengths are that these are the places that are doing iGenetic study, and these are the people which I've listed over here. Who have contributed significantly in terms of iGenetics in India to the global iGenetics knowledge. Now, of course, there are a lot of challenges still. Most of the times what happens in many of the institutions, people do a lot of genetics work, not necessarily iGenetics, but then it's never translated back to the patients. That's a big deficiency. Genetic counseling there are only about less than half a dozen centers in India in ophthalmic hospital where genetic counseling is going on. For example, in Shankar Nethralaya, Aditya Jyoth, Narayan Nethralaya are the places where routinely patients are being seen and then counseled with genetic information. Now, there are two silent contributors, and I have to mention that over here. And these are the people, and I was very lucky to have them as my mentors. Dr. Michael Denton, he was at Otago, and I worked with him, who's on to the left-hand side of the photograph. And to the right is John Fielding Hitchman Check. Fielding is actually, I would say that most of the people I showed, almost about 90–95% of the people trained in iGenetics in India are student of Fielding. Okay, and Fielding is from National Eye Institute. At the, he's still running a very vibrant laboratory in the US, in NIH. Now, what is the actual significant contribution? I went through several of these publications. So this is the most significant contribution by India to the iGenetics globally. So discovery of this RPE65 gene was done both by Shankar Nethralaya as well as Otago University under the leadership of Michael Denton. And at this point of time, when this paper was published, back-to-back Ted Ryder's group in Boston also published. And the difference was that this was autosomal recessive childhood onset severe retinal dystrophy, whereas Ted Ryder's group showed that the same gene is linked to Leber congenital amaurosis, or LCA. Now today, this is translated not only in iGenetics, but in the entire field of medical genetics. This is a successful gene therapy so far done. And the gene therapy is now a product by this company. It's phenomenally expensive. It's about $850,000 US for both eyes. And that is being marketed by this company called Spark, and the product name is Luxturna. And it's currently available as prescription medicine for people with RPE65 mutation and retinal degeneration. And the person who made it possible is this person, who's at the Moorfields Eye Hospital, and he's a colleague of Sharola Dharmaraj, who's over here. So Dr. James Bainbridge is the first person who did RPE65 gene therapy. He came over to us in Narayana Nethralaya, and of course you can see Fielding and Michael Denton, who came about 8 years ago over there. And at the same point of time, Jean Bennett did the same work in the US, and now the company that has, this Spark is actually, she's part of it. So I should thank a lot of people with whom I worked, and together in multiple institutions where I worked, we have actually published more than 60 to 70 publications on iGenetics. So starting from, I should thank the National Eye Institute where I was trained, and you can see Fielding Hedgemansick over here. So this is 1998, okay, so many years ago. And some of the people who you can see, this is, sorry, that's Kapfa, Mrs. Kapfa, who was the wife of the director of National Eye Institute at that point of time, Dr. Karl Kapfa. And this is the time I left Shankar Nethralaya, nearly about 10 years ago. In fact, this is the team, great team. I really enjoyed working with them. And this is the glaucoma team, which was running under the leadership of Dr. L. Vijaya and Shankar Nethralaya. We did a lot of iGenetics glaucoma work. And this diabetic retinopathy work we ran with Dr. Tarun Sharma and Dr. Rajiv Raman in Shankar Nethralaya again. So this is the group currently I'm working with in Mumbai, the Aditya Jyoth Eye Hospital. This is the foundation, which is the Aditya Jyoth Foundation for Twinkling Little Eyes. And this is Dr. Natarajan. Of course, the CEO is Dr. Radhika, who is also in the audience here. So this is the group I'm working in Bangalore, doing very good work. They're setting up the first gene therapy lab in the country, and the person who's responsible for setting this up is Dr. Arka Ghosh, who's over here, and these are the clinicians there in Narayana Nethralaya. Sorry, the slides are not moving. Okay. This is a Chennai unit where we are running a non-profit genetic unit, and we're doing free of cost counseling and genetic services. If people cannot afford, we charge people who can afford. This started by Dr. Vaithlingam Dev in Chennai. So I come to the end of my talk, and this is good contribution done by India to global genetics. And I again thank Dr. John Prakash and Dr. Takeshi Iwata for this opportunity. Thank you.
Thank you very much, Dr. Kumar. We're going to hold the questions, I guess, unless you have any burning question, anyone has a very special question to ask. We're going to hold this until the end. So We've changed the order a little bit. Is Dr. Richard in town here? He's not. Okay, no problem. So we'll go with Professor Sundaresan here, because after Professor Sundaresan we'll do Dr. Iwata and myself. So most of you know Dr. Sundaresan, who has done elegant work and continues to actually be a big contributor to our knowledge, not only in India, but at a very international level. I have worked with Dr. Sundaresan for a number of years, a very prolific group he leads, and so I'd love to welcome you here. Please, sir.
Thank you very much, John. So first of all, thanks for the opportunity to stand in front of you. So the given topic to me is update on the molecular genetics of eye diseases. I'm not going to focus only in India. I'm going to talk about the entire globe, what is going on in the ophthalmic eye genetics. I think if you see the eye structure from cornea to the optic nerve, I think extensively studied. If it is a Mendelian disease, we can easily able to identify the candidate gene for that particular disease. And if it is a complex disease like a cataract or pseudoexfoliation, that kind of thing, what you have to do, it is impossible to collect the sample, only the genetic sample, because there is no, you can't able to collect the positive family members because the age of onset, because the age of the onset is the pseudoexfoliation and the glaucoma and the other AMD like that is a very, very late onset. So people are adopting the association studies. It means collect a huge number of samples and then you can go for NGS analysis. So that's what people are performing, and then they are identifying the candidate gene. So I think this is a paper, very recently published a paper. The 1 to 10 is the major problem of the list of the eye diseases. As I mentioned, they identified the candidate genes, our gene variants, and then the age of onset, the onset of those diseases. It is listed out 1 to 10. And this is another paper very recently in 2017. Suppose I'm focusing a little bit clinical retinal dystrophies, glaucoma like that. This is dealing with towards the molecular genetics of inherited retinal degeneration. Suppose the human genome is shrinking. It means there is no more 25,000 genes. It is only 19,000 genes. They proved, during the, using the antibodies, specific antibodies, and then 19,000 genes are there. Concerned with ophthalmic genetics, only 550 genes are there. So among the I'm telling the approximate number, 550 genes, the back of the eye, like a retinal dystrophies, covering more than 300 genes. Like the 250 genes are there only for the retinal dystrophies. So the human genome is completed, retinal dystrophies are 250 genes. If you take Harvard Medical School, those who are coming to the retina clinic, they are screening the entire gene panel for that particular patient. So if we screen the 260 genes, obviously the particular patient is positive for one particular mutation. So this particular paper, it is very true information. So you consider the patient is, we diagnosed as a retinoblastoma, retinitis pigmentosa or AMD. So known gene is there, maximum what you can do, you can able to identify using the NGS techniques only 60%. That is highlighting in this paper. 40% of the gene mutation, even if it is a confirmed case, it is impossible to identify what is the causative of the genes. That is the highlight of this particular paper. That is proved in many, many countries, from France, UK, Spain, and that is listed out. If you see the percentage in the fourth column, the 64% is maximum they can able to identify the mutation is positive. Rest of them is negative. In the sense, even the negative sample is a gold mining. What you can do, you can collect the known, first you screen, and then if it is negative, you take the negative genes, and then you can go for gene discovery to understand what is going on in the retinal dystrophies. That is the highlight of this particular paper. And then right now, obviously, whatever you are doing in the lab, we supposed to transfer our technology from bench to bedside. The recent technology, this is another paper from molecular therapy using the CRISPR-Cas9. That is a very hot topic right now. Suppose consider one particular gene, one variation. Using this CRISPR-Cas9, you can able to change that particular change. That is possible. So for that, you have to take the fibroblast of the particular patient. You take the skin fibroblast and then make into through 4 genes, you can convert the fibroblast into that particular thing, and then you can change the modification of one particular gene and then gene mutation, and then you can transfer into the patient. That is the advanced technology. People have already proved in the animal model, and then within the near future, they are going to transfer to the human. So I think that's what they emphasize in this particular in the molecular therapy paper. So here they given 3 points. One is they already did using whatever I told you, the induced pluripotent stem cell for CEP290. That is one of the gene for both retinitis pigmentosa and the Leber conjunctivosis. That is possible, and then MAC as well as the RHO. So these are the same kind of technique, whatever I told you, they already performed. Now the strategies developed in this study will prove useful to correcting a wide range of genetic variation in genes that cause inherited retinal degeneration. That is the current information from that paper. And concerned with this particular global genetic consortium, so I think I pulled down some of the information related to what are the future challenges the inherited retinal dystrophies research and diagnostic. What is the future challenges? So now I think I can read the point 1 to 4. How can we determine the causality of the ultra rare mutations in novel candidate gene? That is the key question. And another one is, suppose if we can't able to do that kind of thing, you have to go for collaboration. That's what they are mentioning. So this particular paper is focusing on the European Retinal Consortium, the ERDS. That is that particular consortium. Using that particular consortium, suppose if you want to conclude a conclusion whether this gene is responsible for the particular disease, or we can able to understand the biology of the particular disease, so you can do that using that particular consortium. So I emphasized here in the red point our global IGG consortium. This is the forum. Suppose if you are dealing with many eye diseases, and then you better to share your information or the genetic material through proper channel, and then we can able to identify. So at least we can understand the pathophysiology of the particular disease, and then we can transfer the technique from bench to bedside. That's the way. And then the second one is, how can we identify and functionally test of non-coding variants? So suppose you should not focus only the coding region, that is called whole exome sequencing. People taken only the exome sequencing, but you have to consider the non-coding regions too. I think a lot of that the expression studies Dr. Avata is performing for his retinal dystrophies, he is going to explain that in his presentation. To fully understand the genotype and phenotypic correlation, that is very important in the sense, suppose I will give an example. You take the fundus, and then you are identified a person is having the RP, retinitis pigmentosa. You are identifying a particular gene or RP65 or rhodopsin gene there is a mutation. And suppose you just look into the fundus, what are the exact phenotype is there, and then you can compare to the genotype. But the problem here in the retina is a $1 million question. So suppose what I'm trying to say is, you take the same kind of patient and then same mutation, you coincide all the fundus together, impossible to coincide, yes, this mutation giving the same phenotype. So it means right now the coming up the artificial intelligence. So in the sense for the artificial intelligence, you need 3 things. One is the excellent clinical information from the patient, and then the image, and then the genetic data. You pull all together, and then we can able to come for this kind of genotypic correlation. And then the fourth one, can we begin understanding the phenotypic differences? These are the 4 important points. I think these are the future challenges we have to work on together using the global genetic consortium. And I will move on to the next major problem in glaucoma. I think glaucoma is a very complex disease right now. See, these are the so many factors involved. Either it may be aging or intraocular pressure or hereditary or so many factors involved in the pathogenesis of primary open-angle glaucoma. This is another paper recently published in Eye in the year 2018. So I think they asked a question, are we ready for the genetic testing for primary angle glaucoma? The answer is no, because there are only 3 genes identified, 25 loci are there, but we can't able to come up with this is the candidate gene for the particular, but gene testing, it is a little bit difficult. But the Genevix group, they have a criteria, if the family history is there, and then the onset is more than 35, if in that scenario, you can go for myosin mutation, only 5% of the patients is positive for myosin mutation. You can read but the last line. So here, I think this is not able to going to bring to the community. That is, is not, currently justified. That is the conclusion of this particular paper. And again, this is an animal model. So this particular paper is emphasizing the animal model. So I think, before going to perform bench to bedside, you have to check the biological relevance. So what is happening before going to the human? You're supposed to prove it in either in the mouse model or monkey model. So here we have in our system in Aravind Eye Hospital, Madurai. So we have a HOCA system, in the sense you take get the cadaver eye, and then you can take the entry chamber, and then you can perform, how the intraocular pressure, changes, how it is blocking. You can go put the steroid, how it is increasing the outflow, like that. That kind of studies we can able to we can able to do it through animal studies. So here they are focusing on two kind of animal studies. If you are really interested, please note down the reference, and then you can go through in detail. Because a lack of time, I skip this one. And I think Dr. Gerald, I think he's from US, California. I think he's one of the founder for the in AIOC routinely conducting the eye genetics consortium here. So very often, we will exchange what is going on in US and other places. So this is the topic from his email. So very recently there is a meeting held in Honolulu, Hawaii. So Dr. Genevix, he's emphasized whatever she is performing. She is working on glaucoma, that is myocilin, she is TEK1, CAL1, that kind of genes. These are the main genes that is a causative genes for glaucoma. And if you read the last paragraph, so lastly she mentioned these kind of genes. I think within the 10 years period, she said it believed that we can able to go for the gene therapy using the CRISPR-Cas9. So that's what is emphasized in the talk. And then, so rightly pointed out Dr. Kumar, so the RP65 gene therapy, successful gene therapy, this is the subretinal injection. You take the retrovirus, adeno-associated virus, put the wild type virus of the RP65, and then you could put it subretinally, and then maximum you can go for only 2 injections, and then the patient should be positive for RP65 mutation. Then only the gene therapy is possible. So now I think this is very, very recent paper from clinical experimental ophthalmology from the group from Australia. I think please, this is an excellent paper. They emphasized the active ocular gene therapy clinical trials, what is going on in various diseases, LCA, RPE, X-linked RP, everything, what is going on, LHON, that is the gene therapy is going on in Pascal Palmer. So like that, it's very beautifully emphasized the entire tabular column. Again, lack of time, I'm skipping. Choroidemia, what is the phase 1 trial, phase 2 trial, everything is emphasized in this particular article. So, like retinoschisis, that is also in the phase 1, phase 2 trial, people recruiting sometimes. So what are the adeno-associated virus vectors using for that particular thing? What kind of injection is intravitreal or subretinal? Everything is mentioned in this particular article. Okay, so right now, again, Dr. Kumar already mentioned about this. I'm talking about in Indian scenario. These are the landmark papers published in RP65. So what we did in and around Madurai, so there are a lot of blind school. So those children or the students, they neglected students from because of the LCR RP, they are studying in the blind school. So just we want to know their genome, what is going on in their genome, especially there are 29 genes are there for LCA. We recruited those patients, and then 30 patients. This is done in some 10 years ago. There is no NGS facility, it's very, very expensive. We got the ICMR approval from director of ICMR. We transfer the material from Estonia, and that company, they have the panel, and then we screened the entire, that is called target sequencing. So we come up with what are the mutations happen in the 30 patients, and the gene therapy is possible only for those who are, as I told you, only positive for RP65 mutation. So in our result, I think how many 8 patients are positive for RP65 mutation. It means they are ready to go for gene therapy. For gene therapy, inclusion exclusion criteria is the patient should, the retina should be intact. If there is any tear, is a leakage will be there. Those who are, positive for this kind of, inclusion criteria, so we can go for the injection of RB65 mutation, using the adenoviral vectors. So we made a proposal to ICMR to implement this kind of gene therapy in India. So it is not possible. The thing is, we already, interacting with the Moorfield Hospital to get the clinical grade virus. You should not, put whatever the virus you are making in your lab and put it into a patient. It will complicate major problem. You need a clinical grade virus. So they have the one, and we are trying to implement that kind of thing in India with the help of ICMR. It may happen within a short period of time or within the near future. And then you can ask a question, what is your contribution? That's what Dr. Kumar already mentioned about that. For the past 2 decades in Aravind Eye Care system, so we extensively worked, you take from cornea to the optic now. So these are the, diseases we extensively studied. All my 20 students, 20 PhD students worked in many, field. Either it's a cataract, a POEG, angle closure, like that, albinism, and all this Usher syndrome, everything, like more than 30 eye diseases. And, now what I'm trying to say is we have a spectrum of genetic variation in Indian population. Like, in southern part of India, the consanguineous marriage is high. The people are not migrating to different places. So we can, suppose if you go to the cosmopolitan city, either the Calcutta or Mumbai, I think already the pool is heterogeneous. Many people are coming to the hospitals from different state. It is impossible to come up with one particular thing. Fortunately, in Aravind, we can able to collect the people from one geographical location, and then you can go for genetic study. That's what we did for the past 2 decades. So in addition to that, rightly pointed out Dr. Kumar, so I think we published more than some 95 papers, and we identified some of the candidate gene for some of the eye diseases. Lack of time, so in 2006, we identified the candidate gene for CHED, that is in the 20th chromosome, that is called SLC411, and pseudoexfoliation. So again, the congenital hereditary endothelial dystrophy is a recessive disease. It means grandfather may have the disease, and father may not have, grandson have the disease. It means skip one generation. We got 5 families, and then we identified the particular gene. But concern with the pseudoexfoliation, as I told you, the onset is very, very late. We collected huge number of samples, like 1,500 sample of the pseudoexfoliation. We excluded the pseudoexfoliation with the glaucoma, and then we collected 3,000 age-matched controls. We did the analysis, and then we came up with the calcium channel 1 gene is additional candidate gene for pseudoexfoliation. Again, angle closure, we identified some of the candidate genes, and the pseudoexfoliation there. Like that, we published some 4 papers in Nature Genetics. And I think if I had time, I can explain in detail. Otherwise, I can stop here. Okay. So. Okay, yeah. So I think this is maybe I will wind up. So this is it will go in detail. The thing is, once you identify the gene, I think you're supposed to perform the gene expression studies. In the sense, suppose for a glaucoma, once the gene is mutated, it will form an aggregation. So that is nothing but the extracellular matrix. It is not going out of the cell. So that's what it is building with the intraocular pressure. The same kind of strategy we did for this CHED. Once the gene is mutated, the product, the protein, it may come to the surface of the cell, or it is sit in the cytoplasm. So you can you put the specific antibody, and then you can compare with the patient and the control, and then you can come to a conclusion. So what is the role of that particular mutation or the gene is in the pathogenesis of the disease? So that's what we are explaining using the different techniques. This is our paper, and again pseudoexfoliation. I think see here. So this is one example. I can't able to do it for such a complex disease using 1,000 sample, 3,000 control. I can't do nothing. So I can't able to come up with the conclusion. That's what Dr. Avata would like to explore, like a huge number of samples all over the world. And then what you can do, you can perform your own genetic study in your lab. You can exchange the data. Okay, through clouds, I think you can exchange the data. With a proper concern, you can exchange your data and then you can come to a conclusion, and then we can identify the new candidate genes or understand the specific disease mechanism for a particular disease. Our contribution here is, I mean, as I told you, 1,500 and 3,000 controls. We collected, huge number of samples. See, this is the first gene, second gene, like that. I'm sorry, maybe during the lunch break, I will explain in detail. And this is for the clinician. Maybe this is my last slide. So concern with the, so suppose if you want to, interested in genetics, I think you're supposed to perform this kind of a flow chart, like a patient presentation to the, whether, who is the study subjects we can focus on for the genetic study. If you follow this particular chart, definitely you can channelize, yes, this is the patient we're supposed to focus, and this is not. No, that is, I think I can stop here. And this is a very, very recent cartoon. As I told you, the CRISPR-Cas9. So that is the ultimate strategy. So here there is a gene, and then one particular position we can able to change, and then you can go for the therapy like using the pluripotent stem cells. So I think this is I think you can, this meaning, nobody is perfect, but we are working on that. In the sense, you can take it in any sense, but why I'm telling is nobody is perfect. Our genome, our signature is not, I mean, it is mutated. Everyone having the mutation, at least 300 to 500 mutation is there. So that's what is, nobody is perfect. After the age onset, we are going to come up with some kind of disease, either it's a diabetes or glaucoma. So once you understand your genome, so we can modify your lifestyle, and then you can prolong your life expectancy. Thank you very much.
Thank you very much. Any questions from Dr. Suda? So we'll come back to the this non-coding stories.
Yeah.
I think there has already been some groups who have did the deep sequencing on the RNAs.
Yeah.
But I don't think all this RNAs has been mapped onto the genome correctly.
Yeah.
So currently we're collaborating with RECANT.
Yeah.
Dr. Hayashizaki, who is the expert on this technology called CAGE, which can identify all RNAs, especially in the retina we're currently working on, and map onto the genome,
Yeah.
and also identify the transcription regulatory elements.
Yeah.
So I think this year we will finish this.
Great.
So it will be in public so that you can map the mutation outside of the genes.
Great.
Yeah.
So I think my experience, I think NGS, DNA is not a problem, but the problem is the RNA-seq and microRNA-seq.
But also,
Yeah.
these biologists have to work on this non-coding one
Sure.
by one.
That's what
Yeah.
I think.
And
That
it's
is
gonna
the
be-
priority.
And I think this is going to be the next generation genetics.
Yes.
Yes, it's the RNA.
Yeah. Thank you.
Okay, next we move on to the core of this session is the iGenetic Consortium introductions. And one slide will be shared by both Dr. Jan and myself, and he will present the introductions.
So it's, thank you, Dr. Sundaresan. It's very difficult to go after somebody who is as energetic as Dr. Sundaresan. It's just impossible. I'm just the opposite, actually. So I'm going to kind of slow down a little bit. But as Dr. Iwata said, we clearly introducing GEGC to India, Global iGenetics Consortium to India. We've done it a few times, and I've actually, as I was telling someone this morning, that I've given all my talks and speech in the last 3-4 days at various, the keynote and the inauguration and all that. So I'm going to be actually very brief. I would rather spend time in the discussion. So clearly what's happening here is that India has played very important role in GEGC. And as Dr. Iwata said, that this presentation is actually made on behalf of the current key members, leaders. As you can see, in addition to Dr. Iwata and myself, we have Paul Baird from Australia, and of course, Dr. S. Natarajan, who has been part of from the founding team until today. So let me just give you a quick, very brief introduction. I'm going to cut down on my presentation, spend more time rather in discussion. Okay, so clearly we know that human homeland used to be in Africa and how things have moved, and our approach is to essentially look at the human migration and work on the GEGC studies. We have established collaborations in, of course, Asia. This association used to be called AEGC. All of members who are present at Honolulu, our ARWO meeting, they actually overwhelmingly, all of them unanimously rather, voted to call it Global Genetics Consortium. And we've started to really have not only interest, but also members from all across that humans have migrated. So, of course, North America, there's a great deal of interest. And as you can see here, I don't want to go through the details of this slide, but clearly human migration is a great story. The aims and goal. So still, we came up with this thing at the founding of Global Genetics Consortium, and still the information remains, the goals remain the same, grand vision still remains the same, and some of you have been with us from day one. I remember Dr. Sundaresan and Dr. Kumar, both of you were there when we founded GEGC at ARWO in Orlando, about almost 4.5 years ago. So still sharing genetic information to identify the common disease-associated variants that both of the previous speakers actually spoke about, establishing a system which seemed to be, again, a bed to bedside and bedside to bed story, and both feeding into each other for accurate diagnosis and disease grouping, establishing a system, of course, with the cost-effective analysis. So we just heard a CRISPR story and things like that. But there is really an emphasis on if we can actually make a contribution in science in terms of doing a genotype-phenotype database. So the good news is that some of you were actually in New Delhi with us just earlier, a few days ago, and we did a symposium on GEGC, and we call it Part 2, which took place at Shroff Hospital under the leadership of Dr. Umang Mathur. And the Part 2 is going to be in Mumbai at Aditya Jyoti under the leadership of Dr. Kumar here. So, and I'll be attending there as well. So we have been working on establishing a database. Dr. Iwata is going to talk about it. We also had a keynote address from Santa Tuminia, who is the Deputy Director of National Eye Institute. And she gave a very nice walkthrough through her system, which she was the founder of when she was the founder of iGene that was established at NIH. So in addition, the grand goals still remain the same, supporting and fostering collaboration, collaborating with each other and forming, working with international as well as the regional organization, follow the human migration study, and then organizing these and holding these conferences and symposia, which obviously we are doing it. So it is a multi-pronged approach, not just that we are just working in labs in addition to, but our mandate, our goal is that very grand multi-pronged approach, several fellowships, scientists, visiting scientists all around. Yesterday I talked about the Indo-US program. It's a multi-pronged approach. We have specific to government of US working with the government of India, and then on several in grants, fellowship, as well as ICO fellowship. I announced the ICO fellowship in my presidential address. So it's really a great story that actually takes the grand vision of our GEGC aims and goals. This again, as I was telling you, the first picture obviously is the one in Tokyo in 2014. That's when the story began. This is the team that basically shook hand and said we are interested and we are going to champion this thing. Rest of it, I think at this point, let's just skip it, but we have been very active. India has played a very important role starting in 2015 when we got together at AIOC at the Asia ARWO time, as well as AOAC in Delhi, and that's when the GEGC India was born, and since then we've done many things. So India has played a very active role, but I want to focus on, I want to ask the leaders like Dr. Kumar and Dr. Sundaresan to really focus on the next generation of scientists. That's where really I want to emphasize it and ask that they devote time doing that, or rather we all devote time doing that. Again, same thing, various places, various meetings. So there is a whole group that we have, we are working on. This is your own Dr. S. Natarajan signing a collaboration agreement, cooperative research agreement with Dr. Takeda at Tokyo Medical Center. And that began a story of starting the first GGC lab in India. So I'm looking forward to seeing it. I've been hearing about it. Dr. Iwara has been there a few times, and all of you guys have been working very hard there. So I look forward to seeing it tomorrow when I land in Mumbai. And that's where this part 2 of the symposium will take place. Again, more meetings, more conferences here. So there's been a very, very active group. We meet every year. The general membership meets at the ARBO meeting, and we kind of go around. We'll be also going to APAO in Bangkok, and then a bunch of other things that are going on. I can mention the discussion time. Okay, so again here, as you can see, there are close to now about 30 countries that are participating. The approximately 180 members at this point, and we are in the process of registering this organization. So this is GEGC China. Last year we inaugurated this, and this took place in Chongqing under Professor Ian's leadership. This is a grand success story in China. They are very active, very similar to India. And then I'm thinking about it, Dr. Iwara and I have talked about that there should be a collaboration between the Chinese group, and we should map the Silk Road, and that, again, understand that the human migration slide that we talked about. So, and this year's in May, the GGC China meeting will take place at Zhongshan Ophthalmic Center in Guangzhou, China. This is on May 15th, and May 16th and 17th. Correct? So this all has led to an effort creating and publishing 2 volumes of Advances in Vision Research. Just one in 2017, first volume, and the second volume is last year in October. We are almost finishing volume 3, and we have volume 4 on the drawing board. And yesterday I heard people were talking about volume 5. So I remain excited. It's just a lot of work. Dr. Iwata and I get together. It's right after my dinner time, in the evening, early night, and that's his day starts. So we continue to work on it. And but it's not just Dr. Iwata and my book, it's your book, it's GGC's book. We just happen to be putting it together. So, and there are several people here, whether Dr. Sharola from UK or Dr. Sundar or Kumar, you all have contributed to this, so it's your book. And I think we are making waves, we've done something, okay? So at this point, I'm gonna hand it over to my partner in the crime, Dr. Takeshi Iwata. It's a great pleasure to welcome Dr. Iwata to India, as well as at this. This is really look forward to hearing.
Thank you, John. So I'll take over the little bit more details of the GEGC. So from the day one of the establishment of the AEGC, we have been talking about this sharing. How are we going to share these data for the phenotype and genotype? And we definitely need a database. First, we were thinking about iGenes, but in some of the database in more fields, also called the GeneEye, has been made about 12 years ago, but these database is not compatible with the next generation sequence. The format is slightly old, so we decided to make our own database. And this is based on the Japanese database that are currently using, but reprogrammed completely from Java to Pythons, and then the menus and will be updated significantly. And we would like to call it like GenEye, a very catchy name for these systems. Oh, sorry. You want to? Okay. So this is the Japanese systems, and then the blue part you put the upload the phenotype. You can have the buttons, you have the menu scroll bars. It's default set so that everything can be quickly phenotype can be quickly inputted, uploaded into the systems. And the yellow part it will be genetics, and the red part is also the comments that you can put. Currently, you can just hit the buttons and all the phenotype data will come in just a roll, and you can scroll, you can output as a PDF, and you have a lot of search engines. So currently, it doesn't look good. I mean, the interface doesn't look good, but the quality of this database is extremely good. And then by using these search engines, you get this the samples numbers, that list of samples that you can just click and you get all this. Now currently the Japanese system is connected to a IBM AI vision systems that that sort of deep learn the phenotype and the genotype. And but the problem is the number of data. It just lacks. One country cannot feed enough data to do deep learning. And that is the case for the rare diseases. If you have a common diseases like AMD, diabetic retinopathies, I think you can easily do this within Japan and within any countries. But for rare diseases, to find a family, maybe 10 families for one gene in one nation, it's going to be very challenging. And for AI, you need to collect about 1,000 pedigrees probably. And for that, I think GEGC will serve significantly important as a player. So this GenAI system is basically a phenotype database systems, and it provides authorization of 3 layers by the country, by the institute, and by the diseases. So you'll be comfortable to input all your information until it's, even if it's not published, so that not everybody will be looking at your data. Only the people in progress, trying to publish, will be looking at it. And once it's published, everybody can see it. So that authorization always has been a problem for the JEGs, Japanese Consortium, that somebody would hesitate to put their data because somebody would just take the data. That never happened, but we will have that authorizations. And also, because of this authorizations, institutes will be only shared by particular institution, can be only seen. Also, this is the very important point, and also Dr. Tuminiya also mentioned at the New Delhi's meeting just 2 days before this, AIOS, that the terminology, describing the phenotype have to be very exact and have to be universal. So we are using this human phenotype ontology, HPO terms, that is not applied for iGenes. And then, so this will be a very good database, and it will be compatible for international standards. Also, multiple search engines will be provided so that you can easily extract information to write papers. And then the analysis of the genetic part will be put in as an Excel file. It's a big Excel file, but also the important part will be displayed on the web, on the screen, so that you can do a little search engines and then try to find the gene you are looking for. Also, we will provide you with the tablet mode, that if you go into this database with a tablet mode, you'll have a visual sort of visual mode of just scrolling through the countries, through the data, diseases, through the genes with your fingers to identify what you are looking for. This will be first placed in Tokyo Medical Center, but it definitely can be moved to any place else. So the new database will look like this. This is a dashboard. Here is the country, and this is only Japan now, but if you put your data on, it will show up at the circle. How many country has contributed to this database? Or the institution-wise circle graph. And institution-wise, you have this data. And then the disease and the genetics, and it goes into the mutation. Once it goes to the mutations, and you can do this easily scroll through, and then you can pick. And there will be number 1, number 2 page, number 3, number 4 to add your patients. So you put the general information and the more detailed information, and then eventually put the genetic information into it. So it's a very user-friendly format that has been significantly modified from the Japanese consortium. And this is the total number, and then this will be the list of samples that you'll be looking into it. So you can, this will be a view mode, and there's no information now. There was no photograph, but there'll be information inputted. This database is coming on May this year, and by our vote, we would like to have this running for 3 different diseases: inherited retinal disease, glaucoma, and AMD. And this inherited retinal disease will contain 36 different diseases from of these type. So what's coming on 2019? So we just had a New Delhi meetings, and then and here we're having this meeting, and then also we will have a another meeting in Mumbai right after this AIOS. The database will be up and running by May, and this will contain 36 different inherited retinal diseases and glaucoma and AMD. So if you're working on this type of, especially the inherited type of these diseases, please contact us, and then we will start the upload of the information. Also, we would like to register GEGC in Japan. We're currently working on this. We will be going to APAO in March, and then May we'll have the RO meeting, of course, and also if you're attending the, this will be on Monday, I think 1:15 or something. So if you're attending RO, please come. And we're, as Dr. Jan Prakash has mentioned, we'll be going to Guangzhou to have a second GEGC meeting in China, and this will be, I think it's going to be very great. So I'm just thinking, this is just for the discussions, but this just goes. So currently in Japan, probably if you take one gene, recently found one gene, probably we will have only 1 or 2 pedigrees. Yes. To really understand the natural history of these mutations, I think you have to collect about 1,000 pedigrees, and then really in different ethnic groups. So this is where GEZ really plays, I think. So currently we have 266 genes just for retinal diseases. Can we do this? Okay, so that's the one of the questions. And once we have these 1,000 pedigrees, we can probably sort of say how many patients in the world for each of the disease genes. And we are being contacted with pharmaceutical companies, venture companies, who are trying to make these gene therapy therapeutics. And every time when they come to us, they will ask, how many patients do you have for this gene, or that gene, or this gene? So answering to this question is going to be very important, I think. And also, as Dr. Suda has mentioned, deep learning is going to take a very big play in this, because this is such a rare diseases. The clinicians who are not experienced in these diseases will mistake in the phenotype. So developing a AI system to sort of assist the diagnostics is going to be very important, and especially for the rare diseases. I think GEGC can play a significant role, because one country cannot do this deep learning on these rare diseases. So I think this is a big, good project for GEGC. And of course, I'm gonna just talk. Can I talk a little bit more? Okay. Can I? On the GEGC, we're not just doing the gene identification, but we're doing a lot of functional analysis, and actually came up with some drugs doing this. So this kind of functional analysis, you do need a good setup on the lab, and then you do need the people who are experienced with proteins and then making animal models and all this. So I think we can collaborate, educate each other, try to get the latest information about the latest technology and share this in protocols so that we can quickly move into this functional analysis and then find out the pathway that leads to the disease onset. So I have been doing the, I have been the head of the Japanese consortium since 2018, and mainly we're working on the inherited retinal diseases, 36 diseases. And the consortium is very complicated. We're right here, and then these are the 30 universities here, but also we're collaborating with many institutions, especially as I mentioned about the non-coding RNAs, we're collaborating with the RECAN who are doing the non-coding analysis for the retina. So we're deep learning. That's what the Dr. Hasek Sensei is doing, saying is that we're deep sequencing the RNA of the human retina, and we will map that onto the genome and also identify the regulatory elements on these regions. So for Japanese, we have set up a disease leader who have a experience for each diseases, because not all the doctors are not experienced with all the inherited diseases. So we have the specialist, and he will monitor the incoming data into the database and decide whether this is really this disease or not. If the quality of OCT is not good, if the ERG is not good, he will immediately talk with this person who is inputting the data and then refine the data to get it right. And this is working very well. So for Japan, we have collected 1,300 pedigrees. We have sequenced 2,000 almost for whole exome and whole genomes. And we're getting quite a bit of information. So we are trying to sequence, if it's a recessive, we will try to sequence all the parents, of both parents, and also even with the normals within the pedigree, if you have it, we'll sequence it, try to filter out the real mutation by adding these normals. And then dominant, you'll have to sequence about 8 or 9%, but it costs more. But if you do this, you'll get the answer pedigree by pedigree. Yes, you don't have to compare with something. You can get the mutation by pedigrees. So these are the filtration that we are using. And as I said, 260 gene has been already found. But I think as we move on to Asia, and also we are interested in moving to Africa, and we're interested in moving to South America. So at this meeting already, I have been, and John has been asked, communicating with the ICO members and the president, vice president, to link us to ICO, to connect with the African research group, connect to the South American research group, so that we can really work on these untouched areas. Yes, we have always been communicating with, of course, European groups, and we have a lot of friends in U.S., of course. But our focus is going to Africa, and we go to South America. Okay. So that's why we named it a global IgG. Well, Japan, 2017, we only had 17% of the diagnostic rate, but in 2018, just 7 years, we have increased to 53%. Moorfield detection rate is currently 65. So in just 7 years, we're catching up. So if you do the same thing, we can quickly and even faster coming up. So these are the genes here and how many patients. This is exactly what we want for GEGC. Gene list, how many patients we have, this kind of things. And if one company comes and then they have the therapy for this particular gene, we can say, oh, GEGC has 20 patients or 30 patients, and it can go for clinical trials. The mechanism of this, I think we have to work on this proteins and IPS and knock-in mice, of course, to make a drug. And we have found many genes. This new gene, There's another new gene, and here's another new gene. All of this is a new gene found in the Japanese population, especially this one. It's very interesting. This is a gene that is expressed in bipolar cells, the synapses of the bipolar cell. But if you have a mutation, somehow the photoreceptor is gone. You have a mutation in bipolar cells, but the photoreceptor is gone. So this kind of unique sort of phenotype can only come from a unique population. And I think as we go on to the population of unique, we'll find more and more something that we have never encountered. And that's, I am personally, as a molecular biologist, looking forward to the GEGC in this functional aspects. Also, this is a family of normal tension glaucoma. It's caused by optineurin. And quickly, I won't go through the details. I'm just going to summarize. This, if you have a mutation in this optineurin, there is a protein called TBK1. It's a kinase. It binds to this mutant, only to this binding. So my postdoc thought that if you sort of put the drugs and deassociate this protein, the TBK1 from the optineurin, you might be able to save the normal tension glaucoma. So that's what we started to screen. And by the end, we found this TBK1 inhibitor called amzenonox. It's already in use, FDA approved, and it's for allergic asthma and rhinitis. And this is actually used in my hospital as well. So if you apply this amzenonox, the mutant will release the TBK1. So we made a transgenic knock-in mice of this E50K and showing that it does cause normal tension glaucoma, and apply this drug to this mice. And we monitor for 1 year, and at the end, we have this. This is the normal mice with drugs, and this is the knock-in mice. As you can see, the normal fibers is significantly degenerated. But if you apply this drugs, you can save quite a bit neuroprotections. And this is already in use, so we're going for the clinical trials. And then if this is the data of the peripheral retina, the flat mount. You can see if you apply the drugs, this is almost the same. The neurofiber thickness and the density is the same. So going through this functional analysis, we can easily, by our current database and things, that we can quickly find drugs or therapy for these inherited retinal diseases. So I would like to end with this slide that there are currently many therapy on board, on planning, the drugs, of course, gene therapy, and the transplantation of stem cells or differentiated cells from the iPS cells, also artificial. But I think through GEGC, we can sort of find therapy for the early part of this disease and try to find the drugs. So thank you very much.
Thank you very much, Dr. Iwata. We have about 7 minutes. So two things. We are going to have, actually we have lots of time. Both Takeshi and I are going to be standing outside, and we can take all the time you need to talk about GEGC more. But let's address the burning questions. And I want to also mention that if we could all stand at this stage. Dr. Takeshi loves to have picture, and he loves to put it on the slide. So we can actually use this picture. This is really very nice that all of you are here. So which one should we, let's do the question first. 2 questions we want to take. No, nothing at this point? Yes, Dr. Yes, Ram. Yes.
So what's the effort in India in terms of phenotypic associations? There's a lot of push from Kumar and Sundaresan. Is there a kind of a push to, like in Japan, to Dr. Iwata's beautiful last few slides? Anything like that being done in India? Not really.
Can you go to the
Microphone, please.
Yeah, that's a good question. this is a big problem in India, is that most of the work that has been done in eye genetics are in private eye institutions, and mostly from Arvind Delvi Prasad and Shankar Netralaya. I would say almost 2/3 of the publications, or even more than that, would be in eye genetics from these institutions. So, and until government of India tries to put money and then create these kind of systems where you can exchange both phenotypic and genotypic information, it's difficult to do it. As far as I know, once ICMR tried to create such a system for retinoblastoma, it started off, but it never actually went to the final stage of putting the phenotype and genotype. So it's a big problem that we face in India. There's no proper coordination between the government and the private organizations. The private organizations are much more enthusiastic and want to do good fundamental basic sciences research. So it's a problem. And the answer for your question is that actually there's nothing of that sort at all. Well, the question is that what is actually the challenge and why is it not being done? There were issues, okay, particularly the model that I at least was in, where we tried to bridge a connection between private organizations which were doing work on retinoblastoma and Government of India through the Indian Council of Medical Research. The issue was that the government was not ready to put money into the system, but they wanted the data from the private organizations. So Shankar Netralaya, Arvind Delvi Prasad, all of them took part in it. The key clinician who was doing retinoblastoma work and the lab scientists who were working on retinoblastoma. So I was there, Chitra was there, and a few other people. I don't know who came from Sundaresan's group for that meeting. But the point was that the private organizations were not ready to give the data, both the phenotype and genotype, to government when government was not ready to give them money. So that's where the catch was. Okay. So I'm not making any political statement here, but then that should be done very smoothly, and it should have been a win-win situation. Then it would have gone forwards.
Just a comment here that we have a US Indo program, and this certainly can be taken under that. You'll need to collaborate with a US investigator. Clearly, Dr. Janie Wiggs is working with Ronnie George, and so I'll be meeting with the DBT people actually in just a few days in Delhi. But it would be a very good approach especially for very established labs. Of course, you know, Dr. Sundaresan and Dr. Kumar and several others too. If you want to actually have a multi-center approach that Dr. Kumar is doing at Aditya Jyoti, and has brought several centers actually to work with him. And clearly that sort of thing would be a lot more useful, and DBT actually had indicated to me before that they like the center approach that everybody will feed into that. One of the programs that Dr. Rajiv Raman is doing with all of you, so they have brought several of you, I think 7 or 8 centers, to collaborate on that and put the data in diabetic retinopathy program, which Dr. Sudha Iyengar in Cleveland is doing. So that thing, NIH reviewers really like, and as well as DBT reviewers. You heard from Balu yesterday, Dr. Balu yesterday. So let's stop here so that and we can just quickly take a picture. So please come over to the stage. Please walk carefully up, and yes. Thank you.
Dr. Tara Prasad Das is there. Sir, you have any comment? Or you can come for the photograph.